Hello everyone, so this is group 5 and we will be presenting on the cardiovascular system. The members of our group is Dana Tenaj Smith, Laji Bogle, Christian Chambers, and Amoy Williams. So the objective of the presentation that we will be discussing today includes demonstrating our knowledge of the special structural and functional features of an erythrocyte, explaining in detail the life cycle of an erythrocyte from development to destruction, and listing the functions of erythrocytes and how this changes at different levels of oxygen availability.
Good day, my name is Dana Tanaj Smith, and I will be focusing on Objective 1, which is demonstrate your knowledge of the special, structural, and functional features of erythrocytes. What is an erythrocyte? Erythrocytes are also known as red blood cells and they make up 45% of the blood. In one drop of blood, there are approximately 5 million red blood cells. They lack a nucleus and have a biconcave shape containing hemoglobin for oxygen and carbon dioxide transport between lungs and tissues. Millions of red blood cells are continuously produced in the bone marrow through erythropoiesis, releasing mature RBCs into circulation with a lifespan of approximately 120 days. Following this period, aged RBCs undergo recycling by macrophages in the spleen, liver, bone marrow, and lymph nodes, and this is also called the reticular endothelial system. So they are basically produced to replace old and dead cells. As it relates to the structure of erythrocytes, erythrocytes are notably small, with an average diameter of approximately 7 to 8 micrometers. Erythrocytes have a unique structure featuring a biconcave shape that enhances surface area for efficient gaseous exchange. They contain no nucleus and other organelles. And in addition to this, they contain the protein hemoglobin that carries oxygen to cells around the body. Its small structure also increases flexibility in navigating narrow capillaries. The primary structure of erythrocytes is the cytoplasm, surrounded by a cell membrane. The cytoplasm of the red blood cells is rich in hemoglobin, the protein which is vital for oxygen and carbon dioxide transport. Hemoglobin's acidophilia gives RBCs a red color when stained with eosin in H and E staining. Each RBC contains millions of hemoglobin molecules crucial for oxygen distribution. The cell membrane, which is the lipid bilayer, comprises integral proteins binding hemoglobin and anchoring the cytoskeleton, and also peripheral proteins, which forms a resilient network which enables RBCs to navigate capillaries without becoming ruptured. So, as it relates to the function of erythrocytes, erythrocytes are vital for transporting oxygen from the lungs to body tissues and eliminating carbon dioxide. When they encounter tissues low in oxygen, the hemoglobin inside the red blood cells containing ferrous heme, Fe, that attracts oxygen changes, releasing oxygen. Moreover, carbon dioxide is transformed into bicarbonate within the cell. And finally, erythrocytes are indispensable for sustaining oxygen levels, maintaining bodily balance, and facilitating cellular metabolism. Thank you.
Objective 2, and this is where we will be discussing the life cycle of an erythrocyte from its development to its destruction. The life cycle of an erythrocyte usually contains or involves multiple or several stages, and the first stage being known as hematopoiesis. Hematopoiesis, in other words, is known as the formation of red blood cells, and this usually occurs in the bone marrow, where hematopoietic stem cells differentiate into erythrocyte progenitor cells, which then mature into erythroblasts. These erythroblasts are known to undergo several multiple stages of development in which they start to accumulate hemoglobin. When they start to accumulate the hemoglobin, they then start to gradually lose their nuclei and organelles, transforming into reticulocytes. So the second stage of the life cycle of the erythrocyte is described as the release into circulation. The release into circulation describes the reticulocyte's young immature red blood cells, which are released from the bone marrow and travel to peripheral tissues, which they start to mature into erythrocytes within 1 to 2 days. So, stage 3 of the life cycle of an erythrocyte is usually described as circulation and oxygen transport. So, circulation and oxygen transport, this is where the mature erythrocytes that we discussed earlier in stage 2, this is where those mature erythrocytes are released into the bloodstream where they carry oxygen from the lungs to tissues throughout the body. What also occurs is that carbon dioxide is carried from these tissues back into the lungs during exhalation, and the lifespan of an erythrocyte during circulation is usually or typically 120 days. So, stage 4 of the life cycle of an erythrocyte. So, stage 4, this is where you usually start to discuss the aging of the erythrocyte. During the aging of the erythrocyte, it usually starts to undergo many changes, whether that's being with its function or its structure. It usually starts to become more rigid, lose its flexibility, or it starts to develop surface changes. With all these changes, it usually makes the erythrocyte more susceptible to by removal from reticular endothelial system. Stage 5. So, stage 5 is described as removal of macrophages, and as the name describes, this is where the aged or damaged erythrocytes are removed from circulating by macrophages, usually from the spleen or usually the liver. So now we will be discussing the last stages of the life cycle of the erythrocyte. So we have stage 6, which is described as the breakdown stage, or in other words, homolysis. This is where macrophages break down erythrocytes into phagolysomes, degrading hemoglobin into heme, iron, and biliverdin. Iron is stored in reserves or transported for erythropoiesis, while bilirubin is released into the bloodstream. Stage 7, and this is the excretion and recycling stage. This is where bilirubin is conjugated in the liver to make it water soluble, allowing for its excretion into bile. Bilirubin is then eliminated from the body through feces. Iron released from hemoglobin breakdown can be stored in the body's iron store or transported back to the bone marrow for reuse in erythropoiesis. This entire process constitutes the life cycle of an erythrocyte, from its formation in the bone marrow to its destruction and recycling in the spleen and the liver.
Red cells or erythrocytes are specialized cells that play a vital function in the body's oxygen transfer. The protein hemoglobin, which links the oxygen molecules, may be stored to its greatest capacity due to its special structure, which is devoid of a nucleus and other organelles.
Erythrocytes effectively absorb oxygen molecules from the blood as it passes through the lungs, generating oxyhemoglobin, which is subsequently carried by the bloodstream to all the parts of the body's tissues and organs. On the other hand, by removing carbon dioxide from tissues and returning it to the lungs for expiration, erythrocytes help eliminate this waste product of cellular activity. This procedure sustains the body's metabolic processes and guarantees the preservation of ideal oxygen levels in tissues, finally permitting cellular respiration and general psychological well-being.
My name is Lijay Bogo, and I will be discussing the oxygen dissociation curve and how it is affected by the number of erythrocytes in the body. So, the oxygen dissociation curve is responsible for oxygen transport in the blood. Well, not responsible, but it shows how oxygen is transported in the blood and delivered to the tissues in the body. It depicts the relationship between the partial pressures of oxygen and the oxygen saturation of hemoglobin. This relationship is crucial because it determines how readily hemoglobin releases oxygen to the tissues in need and how efficiently it binds oxygen in the lung when oxygen levels are high. The curve is usually illustrated as a S-shaped curve, and at low partial pressures of oxygen, such as those found in tissues in high metabolic activity, the hemoglobin exhibits a lower affinity for oxygen. This means that it readily releases oxygen to the surrounding tissues. In high partial pressures of oxygen, such as those found in the lungs during inhalation, hemoglobin exhibits a higher affinity for oxygen, facilitating efficient oxygen transport throughout the body. Okay, several factors can affect and influence the shape of the curve, such as pH and temperature. So, the lower the pH and the higher the temperature, it shifts the curve to the right. And that means that it reduces hemoglobin's ability to affinity for oxygen. As I said before, that means that oxygen is released to the tissue. When the pH is high and the temperature is low, hemoglobin's affinity for oxygen is increased, and so the curve is more to the left. This means that hemoglobin readily binds, well, oxygen is readily binded to hemoglobin. Okay, the oxygen dissociation curve can be measured by using the partial pressures of oxygen and the amount of hemoglobin that is saturated with oxygen. This is denoted by the Bohr's effect and the Hill's constant. The Bohr's effect is a phenomenon where an increase in carbon dioxide concentration in the blood can cause a shift to the right of the oxygen dissociation curve. The Hill's constant, denoted as N, signifies the amount of oxygen molecules that is binded to one hemoglobin molecule. How does erythrocytes affect the oxygen dissociation curve? Erythrocytes are able to carry the protein hemoglobin, which is used to bind oxygen and transport oxygen throughout the body. So it plays a crucial role in the oxygen dissociation curve because it affects the oxygen carrying capacity of the body. The number of erythrocytes in the blood can affect the oxygen carrying capacity of the body because when there are more red blood cells, there is an increased potential for hemoglobin to bind to oxygen or oxygen to bind to hemoglobin. And that causes a shift of the curve to the right. And as I stated before, that means that oxygen is released into the tissue. On the other hand, if there are fewer erythrocytes, the oxygen carrying capacity of the blood is reduced because the curve is shifted to the left, indicating that more oxygen will remain bound to hemoglobin and less will be released to the tissues. Yes.
Erythrocytes. Erythrocytes. Erythrocytes silent carriers in red with hemoglobin their oxygen's bed. Their biconcave shape a marveling sight, efficiently ferrying day and night. Life dance a mere 120 days spin, yet vital for the oxygen within. In tissues deprived, they release their whole. Erythrocytes in crimson stories told.
In veins they roam, erythrocytes glide. Though the vessels them narrow in the body still try. We still need oxygen, Thunder asks who might. Cardiovascular, it plays a great part. Big E blood cells kill disease and clear the path of the heart. Clear the path of the heart.
Erythrocytes carry hemoglobin's grace. Hemoglobin binds oxygen in its stem braids. The curve is shaped like the letter S, signaling how oxygen it does possess. To the right it bends, releasing its hold in it as it flows slowly and too. But when to the left the curve does sway, oxygen thinks that within hemoglobin stay.
Released into the world, reticulocytes roam, maturing swiftly, finding their way home. Circulating rivers, oxygen embrace, 120 days of purpose, of fleeting grace. Aging whispers, rigidity calls, flexibility fades, structures fall. Macrophages stand guard in the night, sense and souls vanish from sight.